Yeah.
Good afternoon, sir.
Afternoon.
Sir, within 5 minutes we will start.
मैं दिख रहा हूँ क्या? स्क्रीन पे हूँ। अंकुर सर स्क्रीन पे स्क्रीन दिख रही है, आवाज नहीं आ रही आपकी?
Sir, आपकी वॉइस ऑडिबल है बट वीडियो इज नॉट देयर सर.
एक मिनट। क्या दीपेश?
No sir, video is not there.
Yes. अभी भी नहीं आ रहा है?
नहीं सर, नहीं आ रहा है, अभी भी नहीं आ रहा है.
अरे दीपेश,
नहीं सर, नहीं आ रहा है. No sir, video is not there. Yes sir, now it is visible.
Good afternoon, sir.
हाँ गुड सर, स्क्रीन दिख रही है? शेयर करूँ अभी की थोड़ी देर की बात?
सर अभी थोड़ी देर के बाद में अपन शेयर करते हैं. जस्ट वी विल स्टार्ट विद इन 2 minutes.
No issue.
A very warm good afternoon to all honorable dignitaries, our chief guest, Dr. 𝑉𝒂𝒂𝒏 Kishan Chatap, Honorable Chairman Sir, Dr. Anil Kharia ji, Honorable Vice Chairman Sir, Mr. Shantanu Kharia ji, respected Group Director Sir, Dr. Puneet Kumar Dwivedi, respected Head of Institute Ma'am, Dr. Sapna Malviya Ma'am, respected faculties and all my dear students. I, Ankur Joshi, feel privileged to welcome you all in Impact Lecture on behalf of Modern Group of Institution Indore. This brainstorming Impact Lecture Session 2 is organized under the flagship of MHRD, IIC and sponsored by AICT. The topic of Session 2 is technology transfer from pilot plant to pharmaceutical industry. Now, to continue with the flow of program, I would like to request our Head of Institute, Dr. Sapna Malviya Ma'am, to highlight a brief introduction about the event.
Very good afternoon, Honorable Chairman Sir, Group Director Sir, faculty fraternity, and all my dear students. Seeing to the young leader of coming pharma profession, Dr. Chatap sir, I heartily welcome you, sir. Inspiration, eagerness, and excellency is one of the aspect which we have always seen in you, and we are having this session, and we need a heartening note for you, which is really a essence of research and innovation. We are pleased to host this event sponsored by IIC MHRD. Main theme of our event is pharmaceutical process scale up and technological scale up, which is the need of error. We wish to listen heartening note from sir on this session, and we wish to prosper and spread the need of today's era, which sir has selected the topic, which will definitely lead to the personal excellence with these words. And it's a matter of enormous gratification for me to have such a magnificent, benevolent personality, Dr. 𝑉𝒂𝒂𝒏 Chatap, as a renowned speaker in between us. So I wish you all a happy learning. You will definitely learn many aspects from the sir, because sir has taken most vibrant topic, which is related to pharmaceutical industry, and we all need to move to the collaboration of academia and industry. Thank you
Very much, ma'am, for highlighting a brief introduction on the event. Now, I request our group director, Dr. Puneet Kumar Dwivedi ji, to share some inspirational words.
Thank you, Professor Joshi. Very good morning, in fact, good afternoon to Honorable today's speaker, Dr. 𝑉𝒂𝒂𝒏 Chatap ji, Honorable Chairman Dr. 𝐾𝐫𝐕𝐢𝐯, Vice Chairman Shri 𝐵𝒐𝒏 𝐾𝐫𝐕𝐢𝐯, Head of Institution Dr. 𝒐𝒏 𝐕𝒢𝐯, other faculty colleagues and students, innovators, team members of IIC, Institute Innovation Council of Modern Institute of Pharmaceutical Sciences, Indore. Like just now, we have heard Dr. Sapna Malviya, and she was saying that this is very great initiative from Ministry of Education to empower students and faculties as well for creativity and innovation and promoting technology transfer in the sector specific. In the same context, the IIC Institute Innovation Council of Modern Institute of Pharmaceutical Sciences Indore is organizing this impact lecture. And we are very much honored to have today's speaker, Dr. 𝑉𝒂𝒂𝒏 sir, and all the students are going to benefited from the deliberations in this session, and with the great expertise of Honorable today's speaker, Dr. 𝑉𝒂𝒂𝒏, I hope this session is going to be a vibrant session, and the technology transfer in pharmaceutical sector, students, those who are attending this session, are going to learn things that how they can proceed and how this is going to help them in coming future also. My best wishes to you all. And again, on behalf of Honorable Chairman Dr. Anil Khariaji, Vice Chairman Shantanu Khariaji, and whole modern group of institutions, I welcome Honorable Dr. 𝑉𝒂𝒂𝒏 Chattopadhyay in this forum. Thank you, sir. Thank you for accepting our invitation.
Thank you very much, sir, for your encouraging words. As we all know that we are in the learning phase, and at this stage, what we learn will develop our mind and create personality for our future. So we have very respected, young, dynamic and energetic, renowned personality, Dr. 𝑉𝒂𝒂𝒏 𝐾𝐫𝒏 sir, with us. Sir is a ex-president APTI MP State Branch, Associate Professor in Department of Pharmaceutics, Head Training and Placement Cell at H.R. Patel Institute of Pharmaceutical Education and Research, Shirpur, Maharashtra. Sir has published 16 years of experience in the Department of Pharmaceutics, and sir has published 56 national and international papers in the very reputed journal. Sir has 8 patents, out of which 2 patents are granted and 6 are filed. Now, without taking much of your time, I further like to call Dr. 𝑉𝒂𝒂𝒏 Chattopadhyay sir to address our audience with his valuable words.
Thank you, Dr. Ankur Joshi sir. First of all, I would like to thank Dr. Anil Khariaji sir and his team, especially Puneet sir, Dr. Sapna Malviya madam, and his team of organizing team. And along with that, I like to say that regarding the topic we have chosen for taking a grant from the respective agencies of IICT. It's very interesting topics, and it will be beneficial for the institutions. And specifically, as a one group of institutions, I know very well these institutions have the two industry, pharmaceutical industry, that is Nandini Laboratory and modern laboratories, and really that topics it will be give the benefit of the students. Though the research will be carried out into the institutions levels, which will be transform the technology into the pharmaceutical industry, that is modern group of that is modern laboratories as well as the Nandini laboratories. So really, it is this topic is chosen. It is you can say that is beneficial for the students of the modern group of institutions. So can we share the slide for further lectures?
Yes, sir, you can.
Varsha
Yes, sir,
kindly
yes,
allow sir to share his slides.
Yes, sir, I have already allowed.
Yes, sir. Sir, permission is already given.
Yeah. Yes. So, just starting my today's sessions, I would like to thanks the organizing team members who have given the opportunity for today's session. The cost. So ultimately, that products will be efficient and the good quality products which will be delivered to the patients. So before that, I would like to. That is the basic of should know about that the pilot plant scale-up techniques and the technology transfer and how to deliver the this research. This comes into the customers. And as I have already know that is the basic audience. At the audience point of view, I have delivered the topics. Will be that is the pilot plant, the research from the laboratory pilot plant to the large scale industry. So what do you mean by the pilot plant? It is the place where the money, material, man, method, and machines. That is 5 M should are brought together for the manufacturing of the good quality products. That is the thing about the plant is there. And what is pilot plant? Pilot plant is defined as the part of pharmaceutical industry where the lab scale formula is transformed into the viable product by the development of reliable practical procedures for manufacturing. Example of that R&D, pilot plant and productions. We can say that if we are doing any making of any formula into the lab scale level or the R&D scale, that is in 1x, and that transform that formula from 1x to that is the 10x into the pilot plant, and that 10x formula should be manufacturing into the 100x. That is the thing what the large scale production is. So production batch will be increasing from 1x, 10x, and the 100x. That is the thing where the from any technology should be transferred from the R&D level to the production levels. That is the what do you mean by the scale up and technology transfer? The technology transfer is the art of designing of prototypes using the data obtained from the pilot plant model. First we have to prepare for preparation of any master formula should we need to carry out the raw data where it will be actually products will be prepared. So those mistakes we should find out into the lab scale levels to avoid that, and finally should be prepared the perfect formula which will be that formula should be transformed for the large scale products. So which are the technology transfer agents are involved? First, that is the R&D units. That is universities, institutions, research laboratories, public research centers, institutions, or even maybe a modern group of institutions. That is the part of R&D units. And another that is where it is the technology transfer and reaches up to the patient is there. That is supply of technologies, R&D or third parties, and large R&D departments and supply. That is the industry is there specifically. You can see that. So how to generate the idea? Generally, best example in that any idea should be generated in your mind first. Then that research should be carried out into the R&D level or the lab scale level. That is you can say that is the startup firms or in small scale levels. And that idea should be specifically generated in university or research institutions like modern group of institutions or any other institution. So that the idea should be carried out onto the practical levels into the research institute. Then that research institutes will be develop that products into the from 1x to the 10x. First we have to prepare for the one formula, then it should be prepared for 10 formulation batch. Then 10 formulation batch should be prepared into the pilot plant levels. Then finally it should be comes into the larger company. That is the production, manufacturing, distributions, and market. In here, I would like to give the one example. Everyone knows that is the Byju. Currently in Byju, owner is starting one of, that is the tuition classes, with starting of 10 students or 100 students. Okay. And that 10 students, you can say that that is the, he has taken the experience of that market, or you can say that that research you have to carried out or completed teaching is good, that is in 10 students. That is, you can say that the before that starting he has generated an idea in his mind. पहले उसके दिमाग में एक आइडिया आई होगी कि नहीं अपन ट्यूशन चालू करें, है ना। तो वो ट्यूशन चालू करने के लिए पहले एक ट्रायल एंड एरर लैब स्केल डेवलप, लैब स्केल यानी क्या हुआ कि एक वन रूम दिया होगा, उन्होंने 10 स्टूडेंट के साथ में एक बैच चालू किए होंगे, ट्यूशन चालू। उसके बाद में उसको लगा कि नहीं, अपने एक पायलट प्लांट लेवल पे एक बैच चालू करते हैं। उसने क्या किया, जो 10 स्टूडेंट थे उनको एक इंक्रीज यानी बैच इंक्रीज करके वो 100 स्टूडेंट तक लेके आए। 100 स्टूडेंट तक लेके आए, वो बैच एक सक्सेसफुल हुआ। उसने फिर एक फ्रेंचाइजी चालू की। फिर वो फ्रेंचाइजी चालू करने के बाद में जो पायलट प्लांट, एक पायलट प्रोजेक्ट के बेस पे एक फ्रेंचाइजी चालू की, वो फ्रेंचाइजी जो थी वो सक्सेसफुल, दैट इज पायलट प्लांट इज बिकम द सक्सेसफुल फ्रॉम लैब स्केल टू द पायलट प्लांट, इट इज द सक्सेसफुल। देन ही हैज to comes into the market in large quantity, that is, and now it is in globally, everybody knows that is to transform that technology from the one classroom to the digital platform, and he has studied in lot in the global market and given the technology from one classroom to the whole world is there. That is nothing but the large scale industry. It's starting from the one room, then it is the franchises, that is nothing but the pilot plant, and that is now it's globally given that knowledge, that is in digital format or either in, that is the in physical format. That is nothing but the large scale industry. So that is the example of that how the research from pilot plant to the R&D, pilot plant and the large scale. So types of technologies, we can say that into the pharmaceutical industries have 3 types: emerging technologies, innovative technology, and established technology. In emerging technologies, we can say that it is the technology that currently is undergoing in batch scale testings. That is called as the emerging technology. But in called as, that is the innovative technologies, it is the technology that under the scale up. In, we can say that in innovative, that is only you have in your mind that idea, it is in starting to doing some research onto the lab scale level, that is the innovative idea. But in case of that established technology, we can say that that is already developed, already set up, and already it comes into the market. That is called as, that is the established technology. And emerging technology, that is on in, that is the lab scale levels in pilot plant base. Content of technology transfers, which involve, that is the 6P. First should need, that is the proper research work. Then that research work should need to carried out into the institutions or the laboratory level. That is the proper work you need to do it. Then finally you should need to think about the pricing of that research or work or idea, whether it is, once it is reaches to the people, whether it is feasible to, that is acceptance or not, as per the pricing. That means need to reduce the cost of your product also. Then publicity, then partnership of any pharmaceutical larger industry to it will be help your products to the reaches to the every people. That is the, and finally that is the people acceptance. Your product should be acceptable by the people. So it will be marketing or selling of product is become the more is there. Then the objectives of pilot plant. Why you have to need to develop the pilot plant? Then we need to try the process of model proposed plant before committing the large sum of money onto the production unit. If you investing, that is for the larger large scale, for that purpose you need more amount of money. In such conditions, there will be chances of loss of money also. So first you have to try it on your pilot plant base. If pilot plant base is successful, then you go for the larger scale. So examination of formula to determine its ability to withstand batch scale and process modifications. In pilot plant, what we have done, any formula should be developed into the lab scale levels, should be prepared the 2, 3, 4 or 10 batches, and if and to check the quality of the product, and if it is not you are expected, so accordingly should be modified the process or technology. So finally, once you should be achieve at the technology, achieve the quality as you required onto the lab scale level, then you should be transferred into the large scale level. So it will be helped also to failing of such products into the large scale level. Another, to identify critical features of the processes. So many, it is you can say that critical points. So you should be reduce the cost, reduce the time, etc. Should be that step should be identified into the small scale level. It will be benefit onto the large scale level. Another, prepare the guidelines for the production and the control process on the basis of lab scale levels, or either that is the pilot. Another, to provide master manufacturing formula with instructions for the manufacturing procedures. Means here you have to prepare the one standard operating procedures where we have to going to manufacturing of the that formula into the last scale process. So this will be happen only in, that is in pilot plant levels. If it is your standard operating procedure is successful, so there is no chances of failure of product into the last scale basis. Another, to avoid the scale up problems. So what are the problems? It becomes into the large scale levels, directly goes into the from lab scale to the large scale levels. So such problems can be avoided into the pilot plant levels. So that are the objectives of pilot plant. Ultimate objective of pilot plant is to reduce the cost of your product or avoid the loss of lump sum loss of any money. Reason for building a pilot plant: to evaluate on process of large scale in scale up operations, to find out and examine the all product or the waste products. So you can convert that waste product byproduct into the your that is the useful products. Another, to produce a trial lot of quantities of the material if you go for the large scale purpose. So waste of quantities of the excipient should require more amount, but it is in pilot plant levels should be in small amount of quantity to be find out the expected results onto the pilot plant. In clinical studies, analytical development, process validations and stability settings, all should be carried out into the pilot plant levels, which will be beneficial for the large scale levels. Then steps are involved into the scale up. Development process for formulations, which involve, that is specifically, that is the 4 steps. First, that is the development batch, that is we can say that is the laboratory batch. Then pre-exhibit process qualification batch, we call that is the scale-up batch. Pre-exhibit batch as well as that is the scale-up batch. That we can call that is the scale-up batches. Finally, if it is that batches, exhibit batches become the successful, then we go for the
Ankur sir. Ankur sir. Ankur sir.
Yes, sir. Sir, Ankur sir is not here.
ये सर लेफ्ट हो गए क्या?
No, sir. Sir, yes, sir. Sir left.
सर का सेशन कंटिन्यू नहीं हो रहा है.
Okay, sir. I call sir.
Sorry, due to internet connectivity issue, unable to connect it. So now, the laboratory scale batch is nothing but that is the 1x batch. What is the use of that? To support the formulations for packaging development for preclinical trial basis is very important. The lab scale batch. Another is pre-exhibit batch. In that pre-exhibit batch, called as that is it is the first step into the scale up, and you can say that that is the 70% of the size of the exhibit batch should be preferred for that manufacturing purpose, and is prepared with complete process documentation and fully validated and analytical methodologies. It will be beneficial for that to detect any problem arise in manufacturing of exhibit batch. And exhibit batch, it is nothing but the demonstration batch. Means before manufacturing of the large quantity batch, should be first go for the exhibit batch with full of that is documentations, everything will be involved in that. So we call as this exhibit batch, it is also called as ANDA, that is ANDA, or either that is NDA or ANDA batch. Regulatory reference batches, bioequivalence batch, fully GMP batch, or stability test batch, you can say that. Or you can say that trial and error batch, it's not, it is the final batch or of the large scale level called as. Contain manufacturing documentation, product specification, accelerated stability data. We are going to submit to the FDA, Food and Drug Administration Department. It is sorry for disturbance. It is manufactured and packaged under the full GMP conditions. And another that is the production batch. You can say that that is the it may be carried out either as a shared responsibility between the development laboratories and its appropriate manufacturing counterparts. It is produced during the routine marketing product. Can called as that is the production batch means 100x quantity, which will be more. The actual size of pilot plant production is 100x batch, may vary due to equipment and raw material variability. आपने अभी जो मैंने तीनों बैचेस बताए, आपको चारों बैचेस जो बताए, लेबोरेटरी बैच, प्री एग्जिबिट बैच, एग्जिबिट बैच एंड प्रोडक्शन बैच, इसमें आपको एक ये जो क्वांटिटी आपको दिखाई दे रही होंगी, इसमें जीरो बढ़ते गए. इसका मतलब होता है ये अगर लार्ज क्वांटिटी में आप अगर प्रोडक्शन अगर एक साथ में अगर करते हो, अगर ये लेबोरेटरी स्केल बैच और पायलट प्लांट बैच अगर हटा देते हैं तो इसकी क्वांटिटी बहुत होती है. अगर बैच अगर फेल हो जाती है तो इसका कॉस्ट बहुत हो जाता है, वेस्ट हो जाता है. तो ये बचाने के लिए ये लैब स्केल और पायलट प्लांट, दैट इज नथिंग बट द स्केल अप बैचेस, प्री एग्जिबिट बैच एंड एग्जिबिट बैच नीड टू कैरी आउट. Another that is scientific scale up. Scientific scale up, the scaling up should done in incremental manners, like that, in incremental manners. Processing being validated for each new scale. Equipment should be purchased following similarity. हमने अभी पहले स्लाइड में बताया था कि एक्चुअल साइज ऑफ पायलट प्लांट प्रोडक्शन जो है ये वैरी हो सकता है, डिपेंड्स अपॉन इक्विपमेंट एंड रॉ मटेरियल अवेलेबिलिटी। इक्विपमेंट की अगर अपन बात करते हैं, शुड रिक्वायर्ड जियोमेट्रिक सिमिलरिटी, काइनेमेटिक सिमिलरिटी एंड डायनामिक सिमिलरिटी। सो व्हाट विल बी हैपन इन दैट, जियोमेट्रिक सिमिलरिटी मींस कोई इक्विपमेंट है, अभी 10 फॉर्मुलेशंस के लिए आपने एक बैच बनाई, उसका जो इक्विपमेंट होगा वो छोटे साइज का होगा, जिसका वेसल छोटा होगा, इम्पेलर छोटा होगा, बैफल भी छोटी होगी। लेकिन वही बैच साइज अगर आप 10x में बना, यानी आप अगर 100 के लिए बना रहे, फिर 1000 के लिए बना रहे, तो उस जो इक्विपमेंट आपने छोटे लेवल पे यूज किए, उसी इंक्रीमेंटल मैनर में आपके इक्विपमेंट की साइज भी बढ़नी चाहिए, वेसल की साइज भी बढ़नी चाहिए, इम्पेलर की साइज भी बढ़नी चाहिए और बैफल विड्थ भी बढ़ना चाहिए। ये उसका बेनिफिट, दैट इज नथिंग बट जियोमेट्रिक सिमिलरिटी। So that it will be beneficial to, that is the production of your batch should not be, problems will become avoided into the large scale levels. Kinematic similarity, this required all corresponding point in each systems to have the same velocity ratios, means impeller speed, आपने equipment तो choose बराबर रेशियो के हिसाब से आपने साइज बढ़ा दी, लेकिन उसी इसकी साइज आपको स्पीड भी बढ़ानी पड़ेगी, पिलर की स्पीड भी उसी हिसाब से बढ़ानी पड़ेगी, क्योंकि पहले क्वांटिटी छोटी थी, इसलिए 10 आपको 10 आरपीएम यूज किया होगा। अगर आपको 100 टैबलेट की अगर बैच बना रहे हो, तो उसको आपको 100 आरपीएम पे बैच बनानी। तो स्पीड ऑफ इम्पेलर शुड आल्सो improve, depends upon the material we are going to use for the mixing parts. Another is dynamic similarity. This required equal force ratio to corresponding point of the systems like gravitation, surface tension, viscosity. Example, आपने कुछ अगर formulations बनाया होगा, suspension का formulations या फिर emulsion का formulations बनाया होगा, उसका आपने viscosity चेक की होगी, 10x जो आपने लैब स्केल पे जो आपने फाइंड आउट किया है विस्कोसिटी, वही विस्कोसिटी आपको लैब स्केल भी चाहिए, लैब स्केल में भी चाहिए, वही विस्कोसिटी आपको पायलट प्लांट स्केल अप बैच में भी चाहिए, वही विस्कोसिटी आपको लार्ज स्केल में चाहिए। जो पैरामीटर आपने इवैल्यूएशन पैरामीटर आपने स्टडी किए होंगे, वो पैरामीटर्स विद इन अ लिमिट चाहिए, जो आपने लैब स्केल में वही आपको यानी लार्ज क्वांटिटी यानी मैन्युफैक्चरिंग बैच यानी मार्केट एग्जिबिट बैच में चाहिए। So the general responsibility or general considerations, so reporting responsibilities there, R&D group with separate staff should be required. Then the formulators who developed the product can take into the productions and can provide support even after transition into production has been completed. यानी जो लोग लैब स्केल लेवल में और पायलट प्लांट स्केल अप में इन्वॉल्व थे, उन्हीं को लार्ज स्केल लेवल में जाना पड़ेगा और बैच एग्जिबिट करके फाइनली मार्केट में जब तक बैच नहीं आती उनको इन्वॉल्व रहना पड़ेगा। उसका बेनिफिट ये होता है क्योंकि जो प्रॉब्लम्स स्मॉल स्केल लेवल में आए होंगे वो उन्हीं को पता है, तो वही प्रॉब्लम्स अगर लार्ज स्केल लेवल में आते हैं तो वो ड्यूरिंग द प्रोडक्शन और ड्यूरिंग द मैन्युफैक्चरिंग बैच वो सॉल्व कर सकते हैं। इसकी वजह से जो लोग R&D में इन्वॉल्व थे लैब स्केल लेवल में या फिर पायलट प्लांट स्केल लेवल में, उन्हीं को इन्वॉल्व रहना चाहिए लार्ज स्केल लेवल। So that is the need of that. So to avoid the problems into the large scale levels, or if any problems will becomes, you can be find out and solve that problems within a short period of times to avoid the loss into the large scale. Another space should be required into the pilot plants. That is administration, information processing, physical testing area, standard equipment, floor space, and storage area. All space should be adjacent to each other, should require. So after that, reviewing of that formula into the pilot plant scale levels, a thorough review of each aspects of formulation is important. The purpose of each ingredient and its contribution to the final product manufactured on the small scale laboratory equipment should be understood. The effect of the scale of then equipment or either that is the ingredients those you have to use into the lab scale level should need to take care of that. Also vendors, quality of equipments, quality of the excipients should need to check that. And same quality should be used into the large scale levels, or same vendors should be used for the production for the large scale levels. So it will be get it benefit for the quality of product from lab scale level to the large scale levels. Another raw materials is the one purpose of responsibility of pilot plant is approval and validation of active ingredients and the excipients is very important. Why it is important? Because raw materials used in small scale productions can be representative of the large scale productions. Equipment that are most economical and the simplest efficient equipment which are capable of producing product within the proposed specifications are used. The size of equipment should be such that the experimental trial run should be relevant to the production size basis. If the equipment is too small and process developed will not be scaled up. And where if equipment is too big, then the wastage of expensive active ingredients will be out. So the excipients, that is pharmaceutical ingredients, active pharmaceutical ingredients along with the excipients and the equipment, those we are going to use for the lab scale level and pilot plant scale levels, that manner, same quality of products and same equipment should be transformed from the numerical manners or in the geometric manners, dynamic manner, kinematic manners. So it will be maintained, that will be get benefit to the quality of products. Those you want or those you want to achieve it will be achieved into the last scale levels. Example, that is tablet production line. This is that is pulverizers, vibrating screen, mixtures, drying over granulator mixtures, tablet press, dust remover, polishings, that is packaging, etc. Finally, that is the product itself, the raw materials, etc. Okay. So this is the production lines which will be required into the pilot plant scale levels, and similarly, that is increase the size of that equipment into the large scale levels in that matter, in that scale up manner. Likewise, example of that is the plant of, you can say that is this plant specifically for that sealing of filling of bottles is there. And here that is bottling plants, sealing purpose. Then production rate. As we know, production rate is depends upon the your requirement or demand of products into the market. Accordingly, should be rate is determined and do the production. The process evaluations. Different evaluation parameters are need to check is there. That is to optimize the each and every parameters into the lab scale level, same onto the pilot plant scale levels, and finally that is the large scale levels. These are the parameters like that. The example of that is order of mixing of components, mixing speed, mixing time, rate of addition of example granulating agents, solvents, solution of the drug, etc. Heating and cooling rates, filter size, screen size, then drying temperature and drying time is there, should need to monitor. All that parameters is need to optimize. If it is not optimized or validate, why it is required to validate and optimize? If it is not optimized or validate, what will happen? The quality of products will be hampered. So avoid that. Each time or each batch should be product or develop product should be a same quality products. For that purpose, optimization validations of each and every steps and your equipment should require to achieve the good quality of product into the last scale. Another master manufacturing procedures. The three important aspects. three important aspects is why it is called as weighing sheet, processing directions, and manufacturing procedures is important. As we know, when I was doing 1 month industrial training at Sun Pharma, Halol, Gujarat, under the scheme of capacity building industrial training from PCI. And at that time, I have recognized that data integrity is a major issue for the quality of product or to qualify the US FDA or the FDA is there or WHO. अब हम लोग जो अभी जो स्टूडेंट्स है आप लोग आपका कैजुअल अप्रोच रहता है वेइंग शीट में और कोई इंग्रेडिएंट से वेइंग करने में एक कैजुअल अप्रोच रहता है। आपको जो क्वांटिटी फैकल्टी ने दी होंगी जो रूटीन प्रैक्टिकल में अगर आप सोचे जाओगे तो जो आपको क्वांटिटी दी होंगी सपोज एक्सी आपको 10 mg लेना है, ड्रग आपको 5 mg लेना है, ये जो क्वांटिटी दी होंगी ये क्वांटिटी एग्जैक्ट उन्होंने एग्जैक्ट यानी लेनी चाहिए। लेकिन आप क्या करते हो, नहीं अंदाज एक दूसरा स्टूडेंट ले रहा है उसी हिसाब से अंदाज से ले लेते हो और फॉर्मुलेशन बना लेते हो। ऐसा नहीं करना चाहिए। और सबसे बड़ा प्रॉब्लम क्वालिटी अफेक्ट करने के लिए ये वेइंग शीट है और वेइंग ही इंपॉर्टेंट है। आप अगर क्वांटिटी कम ज्यादा करते हो तो फॉर्मुलेशंस का जो डोज है वो भी कम ज्यादा हो सकता है, फॉर्मुलेशन के जो इवोल्यूशन पैरामीटर्स है वो भी कम ज्यादा हो सकते हैं। इसलिए वेइंग शीट इज वेरी इंपॉर्टेंट। आपको जो लिखा हुआ है जो आपने वे किया है उसी क्वांटिटी वही क्वांटिटी आपको उसमें लिखनी है। जो डाटा शीट रहती है वो वेइंग शीट रहती है, उसमें वही क्वांटिटी लिखनी है, वही टाइम लिखना है। वो अगर नई चीजें करते हो तो इसका मतलब आपका प्रोडक्ट पूरा हैम्पर्ड हो जाता है, क्वालिटी आप उसकी मेंटेन नहीं रख पाते हो। अगर आपने वेइंग में अगर गड़बड़ है तो आपके प्रोडक्ट में गड़बड़ जरूर आए। इसलिए जो आप रूटीनली एक्सपेरिमेंट लैब्स के लेवल में जो आप परफॉर्म कर रहे हो वो आपको सही तरह से करना है। एग्जांपल बेस्ट एग्जांपल है आपको वेइंग करना है। दूसरा प्रोसेसिंग डायरेक्शंस, दैट मींस that is the standard operating procedures is given to each and every steps, so that should need to follow that. Then and only product of each batch should have the same quality you will be achieving. Otherwise it is difficult to develop the same product each and every batch is there. Another is manufacturing procedures. Manufacturing procedures are given to the students accordingly. First you should need to read it carefully, understand it, then you should be go for the production purpose or preparation purpose of any product. That things is very important is this. Product stability and uniformity. That is the primary objectives of any pilot plant. Is your products should be physically as well as chemically should be stable, and hence the pilot plant is representing the final formulations, and manufacturing procedures should be studied for the stability also. And stability study should also carried out to the before lab scale level, pilot plant scale level, and as well as the finished product. So it will be beneficial for the product or the manufacturer to avoid the loss onto the last scale level. Those manufacturing batches should be carried out into the pilot plant scale levels should need to follow the guidelines for the GMPs there. Specifically, exhibit batch should comply all the good manufacturing practices which will be given by the WHO or either that is FDA or US FDA. So equipment qualification, process validations, regulatory schedule, preventive manufacturing maintenance, regulatory process review and revalidations, relevant written standards, operating procedures, use of component technically qualified personnel should require in that, advocate provisions for the training of personnel because day-to-day routine technology has been going to change, or for that purpose persons are working in that industry should need to take the adequate knowledge or updated knowledge from the experts. A well-defined technology transfer systems should have the particularly system should require from how to transfer the technology from lab scale level to pilot plant, and pilot plant to the R&D, pilot plant to the that is large manufacturing units. I know when I was in Sun Pharma, one of the spark industry into the Baroda, some products are came for the technology transfer. Those who are developed into the R&D levels, then the persons are involved into the R&D levels. 3-4 persons are comes into the Sun Pharma, that is where it is actually production is batch is going to prepare. Some discussion is carried out. What are the problems are comes in during the lab scale levels and R&D levels, and how they are that is counter the problems, and that problems are discussed with the involvement of production unit. Those are going to prepare the large manufacturing branch. So such problems will not become into the large scale levels, and how to avoid that. That discussion or healthy discussion will be carried out in between the R&D people and the production unit. So it will be helps to reduce the wastage of product or wastage of ingredients also. In validated cleaning procedures and all orderly arrangements of equipments so as to ease materials flow and prevent cross contaminations. Cleaning all of two, type A cleaning and type B cleaning. It is very important of any new products that comes into the manufacturing unit or manufacturing of the newly products. So it will be do the both the type A and type B type of cleaning should be there. In type A cleaning, that is the you can say that the no need, you can say cleaning with the swap is there, you can say that. Okay. Or no need to if you change the batch, no need to cleaning, further cleaning is there. But if your product is changed, drug is changed, okay, and total batch will be product will be changed, not batch. In such conditions, you need to follow type B type of the cleaning is there. Means the cleaning should be carried out from the with swab, and you can say that complete cleaning, you can say that. So complete washing of equipments and all the materials are involved in that previous batches should be need to clean it properly. Then should be again, that is the validation of that cleaning validation should be carried out. Then you go for the another new product batches test. And from raw material, arrangement of equipment should be in proper manner. From raw material to the final product should be arranged in one flow, so to avoid the cross contaminations of the products. To transfer of technology, like that is the method or process from pilot plant to the large manufacturing plant. Similarly, analytical method should be developed into the lab scale level or pilot plant scale level, should also transfer to the QA department, quality assurance department, into the large manufacturing unit. So method also develop, analytical method should also develop, and that developed method should transfer to the large scale method, that is the QA department. In another technology transfer, in details we can say that what are the objectives of that technology transfer? To offer the guidance and assistance to inventors and the corporations. Another objective is to provide hands-on learning opportunities. Next, to increase the general awareness of the technology transfer, and to accelerate the scale up and cost reduction. That is more important. The scale up should be accelerate for most of the products. Nowadays, what will happen? So many research is carried out into the lab scale levels, university level, research institution levels, but it is not a scale up into the large scale level because they are, can say that, afraid about the loss, or you can say that it will be not possible to sit with the market. So many problems is there. So such problems can be solved into the lab scale levels. Then and only should be those you know very well, that is, if you just directly scale up and go for the large scale levels, the maybe your batch in large scale batch, maybe that is the loss of that. In such conditions, that cost, loss of batch cost will be included into the your future manufacturing basis. So that should be avoid that. For that purpose, should we find the mistakes onto the lab scale level or pilot plant scale levels, and which will be beneficial for the large scale levels. Steps involved in technology transfers. In generally, here that is the first step in development of technology by the R&D. As we have discussed in earlier, that is design the procedures and selection of excipients by the R&D. And first, that is any idea should be kept in your mind. This idea should be comes into the lab scale level in practical procedures, and that for that purpose, whatever requirement, example that is excipients, okay, should require that excipients should use that from the specific specification and the quality should be checked, and that material first need to check it, quality of that product should need to check, then assembled it into the lab scale level. That is nothing but the R&D level, you can say that, or pilot plant level. Then technology transfer from R&D to the production. So for that purpose, so many points should be. That is, first, you have to prepare the one master formula card. Means आपने जो एक फार्मूला बनाया होगा, वो फार्मूला ऐसा फार्मूला बनाओ कि वो उसको 10x में भी चले, 100x में भी चले. लार्ज क्वांटिटी में अगर अपन प्रोडक्शन अगर बैच करते हैं, तो उसका उसमें क्वालिटी जो पायलट प्लांट लेवल में थी, R&D लेवल में थी, वही क्वालिटी, वही पैरामीटर्स वो क्वालिफाई होनी चाहिए लार्ज स्केल लेवल. वैसा फार्मूलेशन अपने को चाहिए. दैट इज द थिंग विद द मास्टर फार्मूला कार्ड. Then master packaging card. Similarly, also packaging can also affect your formulations or quality of products may be hampered. So R&D department should think also not only for the formulation but also think about the packaging also. Then finally, prepare the one master formula. Then after preparation of master formula, that is specification, standard operating procedures. What are the instructions need to given during the manufacturing of any product into the large scale levels? That instructions should need to prepare. Standard operating procedures should need to prepare. What are the specifications? Means what are the minimum criteria? What are the maximum, that is viable criteria? Minimum, maximum, medium and maximum levels should need to prepare that specifications also. Then optimization and production. For maintain the quality of product, each and every batch should be the same. If you require, they need to optimization and validation each and every study, every steps. That means the validation study should require and scale up for the productions. That should be carried out into the pilot plant scale levels, which will be beneficial for the large scale. Another technology transfer documentation part. Whatever the results will be comes out, should need to note down into the document or should have the proper documentation record. and that documentations which will be helpful to find out the problems also, and it will be helpful to solve the problems. So each and every steps those you have to carry out into the research should note down into the diaries, or should note down into the notebooks, or what are the problems will becomes and how they're going to solve it. Okay, that documentations of each and every time should require. हम लोग जब प्रैक्टिकल कोई परफॉर्म करते हैं तो प्रोसीजर उस हिसाब से नहीं, जनरल प्रोसीजर है, लेकिन प्रैक्टिकल प्रोसीजर ऐसी चाहिए कि उसमें कौन से इसमें क्या डालना है, कितना क्वांटिटी में लेना है, इतनी क्वांटिटी ही लेना है, इसी में डालना है, और कब डालना है, कितने टाइम के बाद डालना है, कितना टाइम में डालना चाहिए, ये सब चीजें छोटी छोटी चीजें जो है स्मॉल स्मॉल पॉइंट्स उसमें इंक्लूड होनी चाहिए. That is nothing but a specification and standard operating procedures. Then it will be finally it should be prepared the development report. Development report should include all the points should be covered, as well as that is what are the problems and how they overcome. And finally, that are the quantity of products and that are the specifications should be required in future. That is technology transfer plan should require what are the requirement for the excipient quality in quantity in terms of that is the equipment in scale up manner should be required. That whole plan should be required and give the report to the concerned department. And finally, another that is check and approvals. In approval and checking, that is WHO. What are the requirement of WHO? What are the regulatory guidelines? And what are the USFDA requirement? You need to follow that. That should be checked and approved into the R&D level batch or pilot and scale level batch. Once it is approved, then you go for the further steps. That is technology transfer checklist. Production master formula, manufacturing instrumentations, deciphering instrumentations, dispersing instructions, analytical methods, previous process validations, stability reports, excipient specifications and sources. Active specifications and sources should know that excipient के sources क्या है, specifications कौन है, vendor क्या है, good quality का है कि नहीं है, ये सब चीजें उसमें आने चाहिए. वैसे ही active ingredient. Primary packaging materials specifications and sources. जो आपने primary packaging materials, that means primary and secondary packaging materials. Primary packaging materials are those packaging materials are comes into the directly comes in contact with the product steps, and secondary packaging are comes in contact with the primary packaging materials, not into the direct contact with your product steps. So what are the packaging instructions? Customer any complaint is into the exhibit batch, or you can say that possibility things which type of complaints may come. Okay, or once batch is passed out, then that is the customer complaint, you should be need to take care of that. Then process deviations file is there. During the manufacturing of product, what are the deviations will become each and every steps. Similarly, in analytical deviation files should be need to carry out during the analytical method development and process validations, process development. Then rejection and rework. If it is anything will be rejected, then need to carry out again starting to how they, that is can say that 𝐃𝐫, corrective action and preventive action need to take a listen. Then you go for the again that is batches will be prepared for the corrective action as well as that is the preventive actions. So that 𝐃𝐫 system, that corrective action and preventive actions should be implemented. So avoid the loss of your product and time, use the time consuming process. Then specific manufacturing batch record. आपने जो भी बैच बनाई होगी उसका पूरा बैच मैन्युफैक्चरिंग रिकॉर्ड होना चाहिए. Similarly, that is specific in our cartoons and labels. लेबल का डिजाइन कौन सा होना चाहिए, उसमें क्या क्या चीजें होनी चाहिए, डायरेक्शन होना चाहिए, वो सब चीजें उसमें इन्वॉल्व होना चाहिए, चेक करना चाहिए. So these are the references I have used during these presentations for sessions. And finally, that is thank you. If you have any questions, you can ask. Any type of questions?
Anybody having any questions?
सबसे बड़ी चीज, can I talk?
Yes, sir.
सबसे बड़ी चीज ये होनी चाहिए कि जो इंडस्ट्री की रिक्वायरमेंट है, आप जो स्टूडेंट्स है इन फ्यूचर में आप इंडस्ट्री में, इंटीग्रिटी इज अ मेजर इशू. जो आप प्रैक्टिकल परफॉर्म करते हो वो एक कैजुअल अप्रोच नहीं होना चाहिए. जो आप प्रोडक्ट बना रहे हो वो किसी ना किसी के लिए यूज होने वाला है. वही अप्रोच आपको इंडस्ट्री में होता है. इंडस्ट्री में आप कुछ भी अगर गलती करती हो तो वो उसकी गलती वो पेशेंट को भुगतनी पड़ती है. तो वो ना हो, इसीलिए आपको जो आप इंडस्ट्री में जाओगे तो उसको वो सीरियसली प्रैक्टिकल या सीरियसली आपको वो जो बैचेस कैरिड आउट करना चाहिए और आपका काम करना चाहिए. सिमिलरली ये थिंकिंग आपको लैब स्केल से आनी चाहिए. जो टीचर आपको प्रैक्टिकल देते हैं वो प्रैक्टिकल सीरियसली करने चाहिए. एग्जाम्पल मैंने बताया कि वे है, वेइंग अगर गड़बड़ करते हैं तो आपका प्रोडक्ट गड़बड़ हो जाएगा. आपने पूरा स्टैंडर्ड ऑपरेटिंग फॉलो किया है, सब चीज फॉलो किया है, क्वालिटी यानी इंडस्ट्री की पॉलिसी फॉलो की, सब चीज किया है, लेकिन आपने अगर वेइंग करते वक्त अगर नहीं है तो आपका प्रोडक्ट भी आपको गड़बड़ा जाए. तो ये चीजें, ये कैजुअल अप्रोच आपका नहीं होना चाहिए, बिकॉज यू आर डीलिंग विद दैट इज हेल्थ केयर प्रोडक्ट and that are directly consumed by the customers, and maybe your customers is become you or your that is the relatives may be there or suffer maybe suffer of the your relative also, or you may be also. Okay. So these things you should need to take care of that. The message from that today's lecture is that it should not be casual approach while entering into the pharmaceutical industry. Thank you, sir. Any questions? If it is welcome.
Anybody is having any questions? Now I request Varsha Joharia ma'am to give vote of thanks.
Thank you so much, sir. Am I audible?
Yes.
Okay. Good afternoon, sir. Today, my words are not enough to express my gratitude. On behalf of Modern Group of Institution, I extend a sincere thanks to our eminent speaker, Dr. 𝑦𝒏𝒂𝒂𝒄𝒆𝒄 sir, who spared time from his busiest schedule to grace the occasion. Today, we had an opportunity to share your precious knowledge, and it will definitely help our students in their studies and in future. And I would like to thank our group director, sir, Dr. 𝐃𝐫.𝐕𝐢𝐯, and head of institute, Dr. Sapna Malviya ma'am, to always encourage and motivate us. And lastly, thank you all the students for paying attention. Thank you so much, sir, and have a nice day.
Thank you. Thank you.
Thank you very much, sir.
Thank you for giving an opportunity to interact with you people and the young mind. Thank you.
Thank you.
Thank you. Can I leave?
Yes, sir, you can leave.
Thank you.